on the surface to bind iron, and it brings it into the cell, and so you can see that green outline, a bunch of them. So there's like 10 cells there maybe, and then in the center of each one of those cells, or at least a couple of them, there you see the red globs, and that represents organelles, the Golgi, the TransGolgiNetwork within each of those cells. And so we wanted to look at we're studying this TGN, the red compartment, in relationship to that green compartment inside. You also see these intercellular compartments, like these other green globs inside of that outline, sort of circles or sort of large, it's a cell. I don't know if you guys get where the cell begins and ends, but yeah, you'll see in the next rendition of it.
Just to get back a little bit to the technology, this is more or less the same as we saw at the beginning. This is the X-ray, let's say, of the microscopy. This is a projection. Here we're seeing something very similar to what you would see on the microscope itself, right? Ryan?
Mm-hmm. Yeah, this is what you would see if you look down on a microscope. Right. Like it's a confocal image. It's called. So it's a series of
so this is a series of images. This is what actually the instrument takes. So it goes some level of depth and takes images.
These are the cross sections.
Mm-hmm.
Right. So this is then very similar to what we saw before with the MRI, right? It's just sections.
But these are 10 microns across,
right,
approximately.
Yeah.
So these are much tinier. I mean, obviously.
So if we have that, we can do the projections and we can do the 3D. Every time we have a stack of images, it doesn't matter where they come from. Basically the same to us. And here you see the cells, right? Very clearly. And the interaction between the two proteins that are labeled.
Do you filter your data at all to reconstruct the surfaces, or is
yes.
this the raw data we're looking at?
This is the raw data, but here we are choosing a level of green and a level of red, and that's what we are displaying. So yeah, I mean, it's biased. You need somebody that understands the science to be able to select thresholds that are meaningful.
So you can't. So it's not shown. What you can't see here because there's no label is the top of the cell. So the top of the cell is absent because the green that you're seeing on. If you see sort of that cup, almost like that's the sides of the cell, mm-hmm. the lateral sides, and that's the side facing the blood side of the body of the organism. Mm-hmm. And this is a kidney cell line. So the other side, the top side, would be facing the urine, basically, of what will become the urine. And that part is absent from there. There's no receptor on that side of the cell. So the cell takes iron up via transferrin receptor, which is in green, and that's only done on the blood side of the cell. The top side, there's no. I mean, if there is iron there, it doesn't want it. It's like trying to get rid of it. So that's sort of what's happening.
And so when you're obtaining a data to do the separation between cell walls and the organelles, do you taint them in the microscope? Like how do you actually get the separation?
Oh, so how do you get the green? Yeah, so the green is a tagged protein. So I added green fluorescence protein to it, and the red is a red fluorescent. It's called RFP, red fluorescent protein. There's a whole bunch of them now. There's many of these. So there's 20. This particular cell line has about 12,000 proteins expressed. There's 25,000-ish proteins that exist in like genes. About 12,000 of them are expressed in this cell. So there's 12,000 genes. I've labeled 2 of the 12,000, and you're seeing 2 of them. You can't see all 12,000 at once. It'd be interesting if you could. But we just take a light and put it on. So it's basically, imagine the way I always explain it, if you have a car and you want to just take every Ford Taurus, I don't know, and you put a red light on it and look at it from the satellite from the world above, outer space, and you look down, you could see where all the Ford Tauruses are. And then you could also say, okay, I want to see how those correlate to BMWs, whatever. And you could see, while they're very different, so the Ford Tauruses tend to be in the Midwest, and there's a large amount of BMWs in New York City, and you can sort of make some kind of conclusion based on that.
we're gonna scale up again to the brain.